Good morning, students. Today we are going to learn light reaction in photosynthesis. Actually, we were learning the photosynthesis, and in photosynthesis we learned that there are the two main processes, that is the light reaction and dark reaction. And in the light reaction that takes place in the grana or in the thylakoids, in the membrane of thylakoids, and the dark reaction that occurs in the stroma. So today we are going to learn about the light reaction. The main products of light reaction are the NADPH and the ATP. And this ATP and NADPH are used in the dark reaction for the synthesis of carbohydrate. Okay, so today we will study the non-cyclic electron transport and how this electron transport occurs. We will discuss today, and this is the light reaction. First of all, in the last class we learned that there are the two photosystems. One is the P680, another one is the P700. These are the two photosystems. And you know that each photosystem consists of the many pigments. So P680, there is a reaction center that contains the chlorophyll A680. This is a special type of chlorophyll A that traps the energy up to 680 nanometer. Likewise, in the photosystem 2, there is the reaction center. That reaction center consists of chlorophyll A700 that trap the energy, light energy, up to the wavelength 700 nanometer. So these are the two photosystems, that is the photosystem 2. This one is the photosystem 2 and this one is the photosystem 1. And these photosystems are present in, this is the thylakoid and this is the membrane of the thylakoid, and this inside, this is the lumen or space of the thylakoid, and this is the membrane of thylakoid. Okay, and this, first of all we will talk about this photosystem 2 that consists of a reaction center, and the reaction center contains the chlorophyll A680 because that chlorophyll A can trap up to the light energy of the wavelength 680 nanometer. So this is the photosystem 2 that consists of one chlorophyll A molecule, and the other chlorophyll and chlorophyll B molecules are also present. Besides, there are many carotenoids are also found. So different many pigments forms this photosystem 2. Likewise, this photosystem 1 also consists of the different pigments that contain chlorophyll A, chlorophyll B, as well as the carotenoids. So suppose now we are going to start this non-cyclic electron transport from this photosystem 2, and then we will discuss about the photosystem 1. So this photosystem 2 that consists of the many pigments, in which the one chlorophyll A680 forms the reaction center and the other pigments forms the accessory pigments. So these all accessory pigments like the chlorophyll A, chlorophyll B and carotenoids trap the light energy. Suppose this is the sun, and now suppose this is the sun rays coming here. This sun rays also going to this photosystem 1 also. So this photosystem 2 and photosystem 1 both trap the light energy. But first of all, we will discuss about this photosystem 2. So that consists of the many pigments which trap the light energy of a specific wavelength. There are the different carotenoids, chlorophyll A, chlorophyll B, that trap the light energy of different specific wavelengths. And all these pigments provides the light energy, trapped light energy to the reaction center, that is the chlorophyll A680. So when the energy comes to this reaction center, this reaction center or this chlorophyll A comes in the excited state from the ground state, and due to this excitement or excited state, the electron now jump from the outermost orbit and this electron immediately trapped by the thiophytine, and that thiophytine provides that electron to the quinone. So what we learned that here, that this photosystem too, now the different pigments trap the light energy and provide the energy to the reaction center, and this reaction center that consists of chlorophyll A680, it comes in the excited state, and due to this excitement the high energy electrons now comes out, jump outside, and that electron first trapped by the thiophytine, and that thiophytine provides the electron to the quinone. After that electron transport occurs, and that electron now passes through the different molecules from this quinone to the photosystem 1, that is called the electron transport chain. This is the electron transport chain that we will discuss. So and simultaneously the energy that also trapped by this photosystem 1 also. Photosystem 1 also consists of a reaction center and the different accessory pigments that trap the light energy of different wavelengths and provide the energy to the reaction center that consists of chlorophyll A700. Here also the chlorophyll A also comes in the excited state, and high energy electron also escape from the reaction center, and that taken by this acceptor. This is the what I wrote here, that the acceptor that accept that electron and provide that electron to the ferridoxine. This is the ferridoxine that provide. So now we start this non-cyclic electron transport from here. Actually, this quinone now provide that electron to the plastoquinone. Actually, this plastoquinone is not the electron carrier, it is actually the hydrogen carrier. So now this plastoquinone also receives the hydrogen proton from the stroma. This is the stroma, and this side this is the lumen. This one is the lumen, or you can say that the space in the thylakoid. So this plastoquinone also receives the hydrogen proton from the stroma, and it forms the plastohydroquinone. And now after that this from plastohydroquinone, this hydrogen proton comes in the lumen or space of the thylakoid here, and the electron now passes to the electron transport system. So if from the stroma, this is the hydrogen proton that receive this plastoquinone, and from plastoquinone you can see that here it forms the hydrogen, and now this electron it forms the plastohydroquinone. Again it lose this hydrogen proton in the lumen side, and the electron now passes to the electron transport chain. So this electron now passes to the cytochrome complex or cytochrome F. From the cytochrome F, it passes to the plastocyanin, and finally this electron is taken by the photosystem 1. So what we learned that the proton from the stroma now comes in the lumen of this thylakoid. So this is the proton, the hydrogen proton now accumulated in the lumen of the thylakoid. So this is the electron transport chain. Now from the photosystem 1, there is also the simultaneously the light energy falls on this photosystem 1 also, and the different pigments of this photosystem 1 now trap the light energy of different wavelength, and this photosystem 1 or the reaction center that consists of chlorophyll A700 also comes in the excited state, and now the electron escape from the reaction center, and that electron now trapped by the acceptor. This is suppose this one is the acceptor, and it provides the electron to the ferridoxine. Now after that, from the ferridoxine, now the electron taken by this NADP, you can see that this is in the oxidized state. So now electron, this ferridoxine now provides the electron to the NADP for the reduction of NADP to form the NADPH plus H plus. So hydrogen hydrogen proton, proton comes from the stroma and electron comes from the photosystem 1, and now this NADP reduced to NADPH plus H plus. So for the reduction of this oxidized NADP, electron comes from the photosystem 1 and the proton or hydrogen proton comes from the stroma, and in this way it forms the NADPH plus H plus. So finally what we learned that for the reduction of NADP, you can see here that this is the oxidized state NADP. For the reduction of NADP, electron comes from the photosystem 1 to form the NADPH plus H plus. And for the reduction of photosystem 1, we can see that the electron coming from the photosystem 2 means the photosystem 2 provide electron for the reduction of photosystem 1, and the photosystem 1 provide electron for the reduction of NADP to form the NADPH plus H plus. Likewise, now for the reduction of this photosystem 2, electron comes from the splitting. Now you can see here that there is the splitting of water. So photosystem 2 also associated with the oxygen evolving complex. Oxygen evolving complex, where in presence of light energy there is the splitting of water, and the water breaks to form this electron, proton, and this is the oxygen, and to form the one molecule of oxygen there should be the splitting of the two molecule of water. It forms the electron. This is the hydrogen protons, and now this is the oxygen molecule. Oxygen molecule goes outside, and now this electron, this goes to the photosystem 2. Means the photosystem 2 get electron from the splitting of water molecule. For the reduction, means for the reduction of photosystem 2, electron comes from the splitting of water, and then this photosystem 2 provide electron to the photosystem 1 for its reduction, and then photosystem 1 provide electron to NADP for its reduction. Or you can say that for the reduction of NADP to NADPH and H plus, electron comes from the photosystem 1, and for the reduction of photosystem 1, electron comes from the photosystem 2 through this electron transport chain, and for the reduction of this photosystem 2, electron comes from the splitting of water molecules. So this photosystem 2 actually present inside of the thylakoid membrane, while photosystem 1 you can see that it is present outside of this thylakoid membrane. So, and this oxygen evolving complex associated with this photosystem 2, and in this you can see that this is the evolution of oxygen, and now this electron now comes to the photosystem 2 for its reduction. Now the question is, this hydrogen, this hydrogen actually temporarily accumulated in this space or lumen of the thylakoid. Means the hydrogen proton comes from the stroma also through this plastoprenome, and the hydrogen proton also comes from the splitting of water also, and this hydrogen proton now accumulated in the space or thylakoid membrane, and this hydrogen proton helps for the synthesis of ATP, that we will discuss in the next class, that how the ATP synthesis occurs or photophosphorylation occurs. So due to this proton gradient in this thylakoid membrane, the ATP synthesis occurs, that we will discuss in the next video. So what here we studied that this photosystem 2 that present towards the inner surface of the thylakoid membrane, and oxygen evolving complex associated with the photosystem 2, and here there is the continuous splitting of water or photolysis of water, we can say, that occurs in the presence of the oxidase enzyme that helps in the oxidation of these water molecules. So oxidase or water oxidase we can say, photolysis of water we can say photooxidation of water we can say, and this photooxidation of water provides the electron for the reduction of this photosystem 2. And here we learned that for the evolution of one molecule of oxygen, we require the splitting of these two molecules of water. And so see this hydrogen proton that temporarily accumulated in the space or lumen of the thylakoid, and this electron comes to the photosystem too for its reduction, and after that this electron now received by this quinon through the thiophytin, and then this electron now moves through the electron transport chain that consists of this clastoquinone, cytochrome F, clastocyanin, and clastocyanin finally provide the electron to the photosystem 1. So here what we learned that actually this quinone provide this electron to the, not the electron carrier, actually it is the hydrogen carrier that forms the electron comes from this quinone, hydrogen proton from the stroma, and it forms the plastohydroquinone, and again this plastohydroquinone provides this hydrogen proton in the lumen or in the space, and the electron passes through the cytochrome F complex and plastocyanin and this photosystem 1. So what we learned, for the reduction of this NADP plus, hydrogen proton comes from the stroma, electron comes from the photosystem 1 to form the NADPH plus H plus, and here hydrogen proton also accumulated here in the lumen of the thylakoid also. Now one thing we learned that for the synthesis of this NADPH plus H plus, how many electrons and protons are required? So now you see, suppose this is the oxidized NADP plus. So for the reduction of this NADP, first of all, it requires the one electron to change into NADP. First of all, it requires the one electron, it requires to change into NADP. After that, to form the NADPH, what it requires? Actually, to form the, first of all, one electron is required to change this NADP into NADP, this you can see. And after that, to change this NADP into NADPH, how many electrons and protons are required? So actually, it requires the one electron and one proton also. This is the one electron and one proton. Electron comes from the this photosystem 1, proton from the stroma. And now to form the NADPH plus H plus also means it requires one more proton also. Means for the synthesis of NADP plus to NADPH plus H plus, it requires the two electron and the two proton. One electron, here we can see that NADP plus, this is the oxidized state. One electron requires to change into NADP. After that, to form the NADPH, it requires one electron and one hydrogen proton. Then it will form the NADPH, and again to form the H plus, it requires the one more proton also, hydrogen proton. Means to change this NADP into NADPH plus H plus, it requires the two electron and the two proton, and that electron comes from this photosystem 1, and the proton or hydrogen proton comes from the stroma. So in this way, today we studied that this is the non-cyclic, because there is no cyclic flow of electron, that the electron comes from this splitting of water, comes to the photosystem 2, then photosystem 1, and finally it forms the NADPH. Means this electron do not return back to this photosystem. So we can say that this is the non-cyclic electron transport. So this is one of the mechanism of light reaction. In this reaction, the product is the ATP and the NADPH. So in the next class we will learn that the production of ATP, that how the production of ATP occurs. Today we studied the synthesis of this NADPH, and this NADPH now used for the reduction of carbon dioxide to form the carbohydrate. So in this way, today we studied this non-cyclic electron transport. Thank you.